Good morning, everyone. Thank you for joining us. First, I would like to thank Alison Walton from the Mongi Center for her help and cooperation. Today's program offers 1 CEU for pesticide license renewal and 1 CEU for certified crop advisors. If you need CEUs, email me your name, email address, and license number. I put that information on the chat box. We are all looking for a cure to citrus greening. I hope we are very close in achieving this goal. Today's seminar program will focus on citrus greening, or HLB, as an immune-mediated plant disease and its applications in HLB management. Our guest speaker is Dr. Nian Wang. He is a professor in microbiology and cell science at the UF-IFAS Citrus Research and Education Center in Lake Alfred. Dr. Wang, please go ahead.
Okay, good morning. Can you hear me okay?
Yes.
Okay. Okay, in today's talk, I'm going to mainly cover these three sections. So I'm going to talk about, HLB is an immune-mediated plant disease and its implication in management. And also going to talk about, our first non-transgenic CRISPR-edited canker-written Hamlin sweet orange. And also I briefly touch the trunk injection. So mainly the first part will focus on the paper we just recently published in early this year. Okay. Okay, so the take-home message is how does CLas cause damage to citrus? Okay, so citrus HLB is with our main conclusion is a pathogen-triggered immune disease. The HLB pathogenicity mechanism really mimics that of the human sepsis disease induced by different microbes. In this for this system, normally the overwhelming immune response is the main cause of disease damage, rather than the pathogen, pathogen factors. So we see the CLas stimulate the systemic and chronic immune response in phloem tissues. Okay, then the systemic and the chronic immune response cause the systemic cell death of phloem tissues. Okay, this cell death really mainly caused by the excessive and chronic production of reactive oxygen species triggered by CLas. Okay, simply we can see the CLas cause the excessive and chronic production of ROS, then cause the cell death of phloem tissues, then cause all the symptoms. Okay, how does CLas cause damage to citrus? Okay, many excellent studies have been done before. They suggest, give them all kinds of suggestions regarding how CLas causes HLB symptoms. Was rejected, suggested like root decay, chloroplast disruption due to excessive starch accumulation in the placentate and the phloem blockage. Due to callose deposition of the phloem proteins or differential transportation of sugars. Now, our current study suggests that the previous observation really the consequence of CLas infection rather than the root cause of the HLB disease. Okay, that means if we try to control HLB by directly, manipulate the root decay, directly manipulate the callose deposition or starch accumulation, you probably will not help. Okay. So I just steal one cartoon from a website. If you diagnose it wrong or misunderstanding of the disease, you basically will give the wrong prescription to the patient. Okay. I hope we found the right diagnosis, understanding the HLB better can give you a better prescription for the HLB management. Okay, so what can we do now? Okay, so I hope you're following me, right? RS seems to be the key, right? So what you can do, right? You can reduce RS damage caused by CLas by increase plant growth and productivity. You can promoting tolerance to RS damage that will increase plant growth and productivity. So there are many ways to reduce the RS damage or increase tolerance to RS damage. The balanced nutrition. Why is that? Because nutrition deficiency causes more RS production. Application of micronutrients, because many micronutrients, at least boron, iron, zinc, nickel, they all can activate the activity of the antioxidant enzymes. Those enzymes can increase the tolerance of citrate to RS level. But I want to caution you, you should not mix them with the copper. Okay, try to separate enough time, because copper seems to reduce that activity, even though copper alone can increase its activity. And also many plant growth hormones or immune hormones. Gibberellin acid seems to be excellent hormone that can protect cells against RS damage, inhibit RS production, and promote plant growth hormone, and reverse RS induce plant growth inhibition. Or the other factors that increase RS damage, in addition to that caused by CLas, heat stress causes excessive RS production. And the salinity stress, the level of RS also increase, and the drought also increase RS production. Basically, our CLas already increased the RS level to unsustainable level. All other stress will further cause damage to the tree. Okay, so what can you test by yourself? Why I see that? Because I've been working with many growers. One of the growers I have been working together with Eddie Liodi. I have known him more than 10 years. Every time I really learn a lot from him. He has been actively testing many different things. Okay, so what you can test, right? So any horticulture, that just again to remind you this, right? CLas, RS, cell death of phloem tissue, and the symptom development, right? This is how we look at this. So any horticulture approach reducing RS damage will help. Any horticulture approaches increasing tolerance to RS damage will help. Any horticulture approach promoting plant growth will help as well. Okay. Another take-home message will be the we have been developing the HLB tolerant resistant Citrus cultivars, where we are. Well, finally, we have successfully developed non-transgenic CRISPR editing technology for citrus. Okay. So if we ask last year, we this technology begin to show some promising. This year we finally have a plant. We know this technology. We can give you the plant. It's a non-transgenic, so really can speed up the application process. Okay. We have generated, we have finally generated the first non-transgenic canker-resistant Hamlin sweet orange using the CRISPR technology. Okay, we also, we have been generating HLB tolerant resistant citrus cultivars. Okay. Okay. So next, I want to talk about what phenotype can we explain using this model, right? How did we reach the conclusion that HLB is a pathogen-triggered immune disease, right? What horticulture approach are working to mitigate HLB damages? Okay. Okay, so what phenotype can we explain, right? Let's see all the known HLB symptoms, right? You have root decay, blotchy model, standard growth of trees. You have starch accumulation. You have hardened leaves. Many things, right? The standard growth, normally when you have an excessive RS level, it's a known conclusion, right? RS actually inhibit the plant growth. Okay. Also cell death of the phloem tissue, right? We know the RS causes cell death of phloem tissues, right? So cell death of phloem tissue will reduce the transportation of photosynthesis, okay, and the death of the companion cells and of the cell element can lead to starch accumulation, okay, then cause the blotch model. The hardened leaves, may result from the RS level directly because RS are known to cause strengthening of the host cell wall, okay. And also, if you go to city growth all the time, right? During active growth time, during later spring and summer, right? HLB symptoms are really not obvious, okay? Then one reason is, excuse me, because we think it is a immune-mediated disease, right? There's a trade-off relationship between the growth and the immunity, right? During the active growth, the immune response to CLas will significantly reduce. Okay, that's one possibility, right? And also, another reason maybe CLas just take time to build up in the young shoots. Okay. Okay, that's some phenotype we think this model can explain. So how did we reach the conclusion that HLB is a pathogen-triggered immune disease? What evidence do we have? Okay. So before I go that, I want to give you a little bit of introduction regarding what is immune. What is the plant immunity? Now, both the plant and the animal have the immune system. Okay. But the shared one, we call it the innate immunity. In the plant world, plant immunity, we also call it plant defense. Okay. So think about this, right? When we talk about the plant immunity, we also refer to basically is a plant defense. But the animal have different one. It have an adaptive immunity, right? But the plants do not have the adaptive immunity. Normally, immunity protects plant and animal against most microbes. That's why we call it plant defense, right? But that's the drawback here. When we call it plant defense, right? You might not think it has any negative effect, but in reality, immune is a problem itself. I know, many of you, autoimmune disease, right? Many, human, we have many autoimmune disease. When the immune was not triggered at the right time, you will have issues. For example, the type 1 diabetes, right? For example, like asthma. They all cause the damage to our body, to our health, really cause our immune system over response. Okay. Now sepsis, right? I have mentioned about sepsis, right? When the microbes get into our blood vessels, that cause our overreaction, our immune response. Okay. Actually, there's many more pathogens. They cause damage by trigger the over response our immune system. Okay. So in the human world, we know host immune system response actually known factor in addition to the pathogen factors from the microbes to cause the damage. But because we call it the plant defense, so we say when really this one has never really triggered the thinking, right, that the plant defense can be issue, to cause damage to the plant, Okay, that's why if you change the term to the immunity, right, you will understand why, the plant and human will have many shared phenomena, right? Okay. So let's again talk about evidence, right? CLas does not contain known pathogenic factors that are directly responsible for causing the plant disease symptoms. This is really different from what we know about all the known plant pathogens. All known plant pathogens have pathogen factors that cause disease. Okay, I will show you, Libobacter does not have any pathogen factors that can cause HLB symptoms. Okay, let's see. Let's see. This is one of my favorite. A picture here. Here's the typical plant pathogen. Okay, this is Xanthomonas. It causes citrus canker, right? I think ovule, plant, citrus canker, right? This citrus canker have many weapons. Okay, this one like a candle, right? It's really called the secretion system. It uses the secretion system to secrete all kinds of pathogenic factors and called the disease. Okay, this one really look like a pirate ship, right? So basically, most known pathogens are like a pirate ship with many weapons, ready to do damage. Okay, actually, CLas is like a yacht, really without any weapons, or maybe known weapons. Leobacter has a very small genome. Okay, compared to its relatives and agrobacterium, like rhizobium, those are those plant pathogens. They have many pathogenic factors like a virgin. Okay, Leobacter really is relative to them, but it has much reduced genome. It lost all those pathogenic factors during the evolution when they live in a phloem-rich environment. Okay. So here's what does Leobacter have? They have the flagella, RPS, pillars. They have a system and a type 1 secretion system, but none of them are known pathogen factor. Okay, so basically, let's give you more evidence. We think they have no pathogen factors. Okay. So basically, what we do is we overexpress all the potential /virulence factors genes in different plants, including citrus, tobacco, and arabidopsis. We see, if they are pathogenic factors, they will cause some kind of symptom. You see, here's the control. Here's the those plants overexpress the putative virulence factors from the CLas genes, but you see no change at all. Okay, then you don't see any symptoms. The plant grew pretty normally. We confirm those plants. So we test more than we test all the potential all the potential putative CLas putative virulence factors. So none of them can cause HLB like symptoms. So this gives us the one evidence that CLas does not contain pathogenic factors that cause HLB symptom. Okay, so now I will talk about more evidence. Okay, CLas infection triggers immune response and cell death in the phloem tissues. Okay, so CLas infection causes reactive oxygen, ROS production, callus deposition, and starch accumulation. Okay, these ROS and callus deposition are the known indicators of plant immune response. Okay. And also, CLas infection causes the induction of the PR genes, another known indicator of immune response. Okay. So CLas causes the phloem cell death. Cell death is more severe in leaves with more severe symptoms. So here I give you some pictures, so you can hopefully see what's happening. What our scientists see. You see, here's the asymptomatic leaves. You see, here's the symptomatic leaf, right? Here you have a high number here means more Leobacter titers. Here's have less Leobacter titers. Okay, you see when you have a Leobacter infection, you do see more. This is a white spot, basically callus deposition. As an indicator of immune response, right? Basically, you have more Leobacter titers, high titers. You have more callus deposition. Okay. And also cell death. Nuclear cell death. See what do we observe? What is cell death? Cell death means the cell die. You see, right? In phloem and the companion cells. You see, very typical here, right? It's around the phloem area and in companion cells. But around the cells, like a parenchyma cell, actually they don't die. It's really only the cells where Leobacter live, they die. Okay, you see the cell death in the phloem tissue, the very obvious cell death here. Here you have a little vector at the early stage, the phloem still intact. Okay. So what the key point we have seen? The cell death in the phloem tissue can occur prior to the appearance of HLB symptoms. Cell death with more severe in liver with more severe symptoms. Some severe element and companion cells undergoing cell death, right? Or others remain intact in the same field because the different infection stage. Cell death was limited to the civil element and companion cell, basically the phloem tissue, okay? But not happening in the surrounding cells. Okay, I have more pictures here. You see here's the asymptomatic, the symptomatic, right? It's basically the symptomatic tissues. You see more cell death compared to asymptomatic, right? Just basically confirming that point I made earlier. Okay, basically, right, we're seeing the cell death of phloem tissues, really the key of the HLB symptom development. Okay. So we want to see next. We want to see how CLas trigger cell death. Okay, phloem tissues. We want to see CLas trigger cell death is caused by RS. You know, normally the cell death can caused by either toxin or effector of the pathogen. You know, I hope I convince you. Now we know the Libobacter, the HLB pathogen, does not have toxin, does not have effector that can cause the cell death. Okay. Instead, you know, another way they can kill cells is probably RS. They're triggered by, you know, immune response. Okay. You know, there's no conclusion here. At a high concentration, RS triggers necrotic cell death, but induces programmed cell death at the lower concentration. So, you know, it's not nothing new here. RS is known to cause cell death. Okay. Let's see whether CLas can trigger the RS level that can kill plant cells. Okay. When we compare when we measure the hydrogen peroxide level, is one of the RS. The hydrogen peroxide the concentration is about 10 to 15 micromole per gram for tissue in a simple leaf, but it's only 2 micromole per gram in a healthy leaf. Okay, right? You see the dramatic increase of RS level. Okay. And also we know when we work with the Xanthoma citrus, right, the canker pathogen, we know when they trigger the hypertensive response, it's another way of another form of the cell death. We know that the hydrogen peroxide level is about 10. Okay, that means CLas can trigger even higher RS level, right? That time that's begin to make us to think. Okay, maybe that's the reason, right? Maybe maybe that's why liver can cause cell death. Okay. We further measure the RS level in the phloem sap. From the phloem sap, in our RS level is about 1.8 millimole per liter. Okay. But it's much higher in the healthy trees. Okay. So in the human study, right? In the animal study, we know at 0.7 millimole of hydrogen peroxide, it can kill red cells, okay, the mast cells. You see, the CLas can trigger much higher RS level, right? That can, let's give us a hint. The RS level can kill can cause the cell death of the phloem tissues, okay? So we test that. But then is that true or not true, right? So basically, we add the let's see, tissue culture, suspension cell culture, the protoplast with different level of hydrogen peroxide, RS. This is the level of the hydrogen RS level we observed in plant, right? Here's the level we in the healthy one. Is the 1.5 that we observed in the HLB positive tree, right? You see at this level, RS clearly can can kill the citrus cell. You know, the green spot means the cells are alive. You know, okay, at 1.5 cell level, very few cells are alive. At 1.8, you know, almost no cell alive now. Okay, they basically give us the direct evidence. You know, CLas cause a high level of the RS that can kill phloem tissue. Okay. So CLas triggers the chronic and systemic RS production. Okay. So we know CLas induce our RS production in phloem tissues. We know not in a normal defense response of most cases, RS production induced by pathogen is really in a very short time, temporary. But in our case, because CLas is infection in the plant is systemic, right? So basically, CLas induces chronic and systemic RS production. You know, we see RS production in the young flush, in the mature leaves, in stem, you know, almost everywhere all the time. So that's why it's the introduction of RS level is chronic and systemic. That's why it's causes the problem. Okay. Okay, the HLB is a pathogen-triggered immune disease, right? We see that RS are the executor of phloem cell death, right? It's directly caused the phloem cell death, but other immune response are also important. Other immune response are also chronic and systemic, including the callus deposition, induction of the immune release genes, right? These all cause damage to the trees, because the continuous activation of the immune system harms both plants and the human. We know the immune system activation is good, but the overreaction and acting at the wrong time is not good. Okay. And we know activation of the immune response suppress plant defense, but suppress plant growth. Okay, that's already known long time. Okay. Okay, when I prepare this talk, Bill called me. I think everybody know Bill from Yara, right? Fertilizer company. He said, okay, you see RS can kill citrus phloem cells. That kill liver bacteria. That's really a good question. I think we do. We do have evidence that RS level can really reach level can eventually kill CLas. Here's the infected tissue. Okay, you see there's a pretty healthy, happy liver bacteria, but when you reach a high level of CLas, you begin to see the CLas are dying. Okay, I think one possibility is CLas dying is caused by the higher RS level. Actually, we have this evidence. Put the CLas in the put the Libobacter crescens, another relative of CLas, into the same level RS. They can also cause death of the Libobacter. Okay. So we see basically to answer Bill's question, right? Hopefully, I think you might be interested as well. So do RS kill CLas? Yes, RS kill CLas. So can we rely on RS to kill CLas then? Can we trigger the RS level to trigger CLas? I think that's probably not doable approach. Because it's a can kill, it's a dynamic process. By the time it kills liver bacteria, it's already caused damage to the phloem cells. So that's why I think it's okay. Okay, other evidence, right? If you want to see now, we want to see more evidence. Liver bacteria, HLB is a pathogen-triggered immune disease, right? If it's such case, right? That means if we suppress RS production, we should be able to suppress HLB symptom, right? Then we should be able to use it for the HLB management. Okay. Again, I want to borrow what happened in our human study, right? We know the antioxidant, immunoregulator, and then nutrition are commonly used to treat the human immune-mediated disease, okay, by halting or reducing RS mediated cell death, okay. Okay, so we do the similar study for our citrus, citrus, okay. So we use antioxidant. Okay, let's see. The conclusion is antioxidant does if it can reduce the RS trigger the cell death. It also reduces the HLB symptom. Okay, let's see right here. I want to see right here's the RS level at 1.8, and it can kill the citrus cell, right? But if you add the antioxidant like a uric acid, right, you can basically prevent the killing effect by the RS, right? So so we did a foliar spray of uric acid on the leaf surface, doing weekly for 6 weeks. We see, and also routine another antioxidant. You see, at 2 weeks, that's what happened. But after 6 weeks, right, you do see the reduced RS level in the uric acid treatment. You also see the reduced cell death, okay, in the phloem tissues. Okay, then you also see the improved tree growth. So basically, the antioxidant can prevent or reduce damage to cells caused by RS. So RS are responsible for cell death of the phloem tissues of CLas infected citrus. We see the cell death of phloem tissues is responsible for HLB symptoms. We see mitigating RS damages, reduce HLB damage. Okay, antioxidant such as uric acid, even though it's so promising effect, it has not been registered for agriculture. But we still don't know whether uric acid, the antioxidant, can be used to control HLB economically. But we can use it to test our concept. Another one, right? GA we know is immune regulator. Okay, it's normally you have a regulate plant growth and also regulate immune response. Okay, let's see if we treat the plant leaves to the foliar spray, we see it can reduce the RS level and also reduce the cell death in the phloem tissues. We also see GA. You see here, right? If we add a GA, we basically prevent the cell killing by the RS. Okay, here's the with the RS without a GA. Here's the with RS and with GA, right? You see, GA can prevent the killing by the RS. Okay, and also work with Alfred. We see in his growth. You see if the GA treated, the plant looks much healthier, right? It's so much less symptoms. Okay, so I want to maybe repeat here a little bit. So GA is a known plant growth hormone and modulate PAM and pathogen-triggered immunity and also the immunity induced plant growth inhibition. Okay, we see if foliar spray of HLB positive trees at 5 milligram per liter and 25 milligram per liter can reduce the RS level and cell death, but the GA also induces expression of the gene encoding the RS antioxidant enzyme activities, and also importantly, GA inhibits the expression of WHD. Why? The WHD is the main producer, the responsible for the RS production in response for CLas infection. Okay. We see the foliar spray of GA can suppress HLB symptoms and improve tree growth. And we see the 30% average increase in yield. This is done by my colleague, Tripti, the group. Okay, we see the G really can increase the plant growth, suppress the growth inhibition by HLB. You see, this is after 7 months data. You see the trunk diameter, the tree height, canopy volume with the G treatment, they all significantly increase for Valencia and for Vernia. Okay. Okay, G effect on HLB control alone is not enough. It needs to be combined with optimized nutrient program. Okay, so you need to have a balanced nutrition because the nutrition deficiency causes more RS production. So when application of the micronutrient, you see, we have some data shows copper, iron, boron, nickel, and zinc at the suitable concentration suppress the protoplast cell death caused by RS. But no effect was observed by this micronutrient. But the effect of the these 5 micronutrients, they have additive effect. But the copper seems to have the reduced effect by those 5. Okay, so that's why if you want to use the micronutrient, please do not mix with the copper. Okay, at least leave some. I think I suggest at least 2 weeks window between copper application and other micronutrient application. Okay, not only our lab, but also other colleagues actually show some evidence that supporting that HLB is a pathogen-triggered immune disease. You see this study by a French group. You see what they have done is, you see is the mecicin lime and the Persian lime, right? We know the Persian lime is much more tolerant than mecicin lime. Okay, so they in both plants, right? They have a similar RS level without Libobacter, but with the with the HLB, with the infection by the Libobacter, you see dramatic increase of the RS level as similar as we observed, right? But in the Persian lime, they have a much more higher, this is the ascorbic acid. Okay, this is the one antioxidant. They have APX and catalase. Those 2 are the antioxidant enzyme. You see, after infection by Libobacter, you see dramatic increase, much more significant increase of the antioxidant activity and the antioxidant enzyme activity in the tolerant variety. Okay, that's really supporting our, this really give us supporting, right? You see, and also in like Utah lab, right? Utah Kim Bowman in 2012, they did a study, right? They compared the tolerant US 897 and also susceptible mandarin, right? You see in the susceptible variety, the Cleopatra mandarin, the Libobacter triggered significantly more activation of immune genes, but in the tolerant 897, that's only very few gene activated. Basically, they are not, much less recognition. They don't trigger the immune response. That's why I think that's why one of the reasons potentially they can trigger their tolerance. Okay, next one I want to show you, our first non-transgenic CRISPR-edited canker-related Hamlin sweet orange. I hope you are as excited as we do. I think you are seeing the first non-transgenic biallelic homozygous canker-related Hamlin plant. Okay, right here. Okay. This is generated by CRISPR gene editing. Okay. So we had already contacted John. I think many of you have known John, right? The Florida Foundation, seed producers. Apparently, we need not more than a tree to get going. Okay. So basically, we have successfully developed a non-transgenic CRISPR gene editing technology for citrus. We think this is the approach we can use to generate HLB tolerant resistant citrus varieties, right? We know that if we knock out the RBHD or reduce the expression, we might generate HLB tolerant trees, because RBHD is the main producer of ROS in response to CLas infection. If we can make the plant not responding to CLas, right, as the result of UTA and Kim, right, you'll be able to make the plant also tolerant or resistant. Also, we can enhance the plant tolerance to RS level, right? As the result from our friend colleague, right? If you have high level expression of the antioxidant enzyme, you make the plant become almost very tolerant to HLB, right? So we're doing that as well. Okay, I'm going to use the last couple minutes. I'm going to talk about what we have done in the trunk injection. Okay, I just want to show you some data for the 5-year data. This is trunk injection with OTC, oxytetracycline for 5-year data. You see, OTC at low level, it really do not affecting the CLas titers, but at a high level, the 3 gram per tree, and also 4 gram per tree. The tree age for those trees actually is Hamlin and Swingle planted in 2013. We did the injection in 2016, 17, and 18, 19, and 20. Okay. And we see in all 5 years, OTC a significant increase in yield. Okay. And also we did if we did also streptomycin, we did a similar setup. For whatever reason, streptomycin, even though they can reduce the CLas titer in if at a high concentration, but there seems no significant effect on the yield. Okay. In all the 5 years. At 5 years, you see the OTC is for 5 years. Create another OTC for 5 years. For the streptomycin for the 5 years, streptomycin for 5 years. Using after 5-year treatment, some trees seem to have some effect. Some trees seem no effect by either OTC and the streptomycin. You see here's the control trees. Here's the OTC treated for 5 years. This tree seems looks a little bit better. But these 2 trees, these control trees, the trees received the OTC for 5 years. There's no obvious difference. Similarly, strep also seemed pretty, maybe it's slightly better. But in this case, no major difference. Okay. Okay. So basically, right, for both OTC and the strep, trunk injection, some trees have seem to have a thicker canopy than the control trees, but not always. So now we'll look at the trial 2. This is Hamlin on Swingle planted in 1999. So we did a treatment in 2019 and 2020. Okay. And also OTC and the strep. Okay, we see OTC again can increase the yield. Okay, but the strep did not increase yield. And in the second season, we also see the increased yield by trunk injection with OTC, but not with the strep as well. But then we did another trial. This is the Valencia trees planted in 2016. For this one, we want to say we basically did injection only in 2019, but not in 2020. We want to see if we, whether OTC, the treatment in the first year, can have a lasting effect on the second year. Okay. You see, if OTC at 2 gram per tree, a significant increase the yield, but the strap no effect. But if we stop treatment with OTC in the second year, then you don't see that no lasting effect from the OTC treatment. Okay. But one thing I want to make you aware, as long as you treat the OTC, even at a very low level, you have this residue level at above the allowed level by EPA. Okay, so OTC in a maximum residue limit is 0.01. So in all the cases, it's above this limit. Okay, in the strap, actually is lower than the limit, but the problem is OTC strap seem not have effect on the yield. Okay. Okay, I want to take home message. Okay, again, right? We hopefully I convince you. HLB it is a pathogen-triggered immune disease. Okay, we think the CLas causes the excessive and chronic RS level. This one causes the cell death of phloem tissues, then causes the HLB symptom. Okay, we see what we can do now. You see, right? We can do all the ways, reducing RS damage caused by HLB, increase plant growth and productivity, promoting tolerance and to RS, right? We have different ways. Balance nutrient, how you apply the micronutrient. I want to make sure I don't mix copper with other micronutrient on the GAA level, right? I think my colleague Tripty has done a lot of excellent work on that regard. And also make sure heat stress, salinity stress, and drought stress you try to be avoided as much as you can. And also what you can test by yourself now, right? So anything you can reduce RS damage, anything you can increase the citrus tolerant to RS damage, anything you can promote the plant growth. Okay. So regarding the HLB tolerant return citrus cultivars, so we we have this technology finally worked out, right? In terms of HLB tolerant return varieties, it's not quite ready. I think we are getting there, because I think we have the technology ready. I think we have the right target as well. Again, I want to hopefully convince you, right? The right prescription comes from better understanding of the disease, the pathogen, the citrus, and the psyllid. Okay. Now, with that, I think really thank you for the funding support of the Citrus Initiative, CRDF, USDA, the federal grant, NSF, and University of Florida, and my collaborators. I think even though I know it's such a long process, but I think finally, I hope we will come out with the solution as soon as possible. Okay, with that, I'm ready for questions.
Thank you, Dr. Wang. We have a few questions in the chat box. The first one is, what's the best way to apply micronutrients, by fertigation or by spraying?
Okay, let me see. Which question you want me? Okay, let me see. You want me to read? You want to read, or I look one by one. Here's one question from Ricardo. What's the best way to apply macronutrient, by fertilization or by spraying? That's a good question. I think foliar spray seems to working pretty well, because the our level seem to be mainly in the leaf, in the stem, in the leaf. If you apply for the macronutrient by foliar spray, they seem to have a direct effect right there on spot. But I think if you do the fertilization, then it might work as well. But of course, the concentration will be different. My only suggestion would be that try to avoid the mix with copper. Here's another question from Matthew. Can you elaborate on not mixing copper with other micronutrients? We rely heavily on copper spray during the summer to control fungal pathogen. That some research trigger the RS production, and then the literature can recommend that. That's a good question. This is what do we do, right? If we mix several the 5, boron, nickel, zinc, MO, these 5, together, we see that actually the effect in terms of the inhibit RS triggered cell death. This is the effect additive. You increase the effect of each other, but you add a copper that reduce the effect. Effect. That's why we only have this data. But in reality, when you apply on the foliar spray, it'll be a little bit different. Well, I really don't have any data in terms of what's the ratio, but for now, I think just try, if you can try to avoid that. Yeah, maybe in that case, right? I think one way to avoid that is maybe, I think if you as long give a couple weeks window, maybe that'll work. Of course, I know that will increase your production cost. Yeah. Okay. Let's see another question. Can you apply copper by
Dr.
leaf?
Wang.
Okay, go ahead, Peter.
I'm sorry. Yeah, great presentation, Dr. Wang. On the CRISPR, you said you're having great success, and we're not quite there. So what do we need to do? What do you need to do to get over the finish line with the CRISPR trees?
Okay. We got the first canker trees, right? We have been working on canker from we got the target in 2014. We finally got this technology figured out in, let's see, last year, the non-transgenic, right? It took us like 7 years. I'm sorry it took so long. But we really began to know the target of the HLB, let's say this year. So we are working on non-stopping to generate that. So I cannot say we have the tree. We are working on it. I only after confirmation, we can only tell you we have the tree. Even that, we still need to test all the, whether you have any negative effect on the tree. So that's why it takes longer. I think it's a number game. You do more, right? You don't want, because we know this is why we try to reduce the immunity, but you do not want to totally remove the immunity, because immunity is really important. RS is very important for the immunity against other pathogens. So we want to tame down the RS level to a certain level. So we have to do, many things. See which at the end, which one is best, right? Have can make it plant tolerant, but not affecting other issues. And also we try to, if we have any HLB specific receptors, I think we have some candidate, we have to test out. I think we feel have something there, but before I get to the tree, I cannot say anything, right?
Okay, that makes sense. Great work. Thanks.
Thank you, Peter. Another
Dr.
question.
Wang.
Okay, go ahead.
This is Wayne Simmons. A little bit more on that CRISPR. The Hamlin with the canker resistant, it's available now for production and out to the industry, so no regulatory issues there.
That's a good question, right? I was really excited when I get a tree, right? I didn't contact John. Okay, so how we get it to outside to the grower? It seems we have to get more data to go through the regulation process. Even though it's not regulated by USDA, not regulated by the federal level, but we still need to make sure this plant have no negative effect on the horticultural trees, like no effect on the yield, no effect on the orange juice. you know, we think so far we did not see any effect, but before we get the data, we have to go get the data. Yeah.
Okay, and so what's your opinion then if you get the HLB resistant CRISPR? Is that going to be a non-regulatory issue as well, or we just have to wait and see?
The way we generate, right, is to be non-transgenic, so it's a knockout. We have no transgenes on the plant, so will be not regulated by the federal level. So, but still, we need to collect all the data. Even I have the tree today, we still need to collect data. You know, the tree will not have an effect on other things, like, you know, tree growth, you know, the orange juice quality, that kind of stuff. Okay.
Okay, great. Put all your resources in getting there.
Thank you, and we're really working hard on that. Yeah, almost all my people working on that regard. In that regard. Okay. Here's another question from the chat box from Ricardo. Can you apply copper by leaf and micronutrient by soil in the same time? That's a good question. That's why I think it might help a little bit, because it takes some time before they can come together. But if you want to apply the copper by leaf, apply the micronutrient by soil, you know, in that case, I think you can make a time a little bit separate, right? Because you don't have to be applied at the same time. Okay, here's a question from.
Be careful when applying micronutrients to the soil. You have to watch really for the soil pH. If you have a calcareous soil or the soil pH is high, then micronutrients won't be available to the plant.
Thank you, Mongi. Yeah.
You're welcome.
Yeah. The Uta had a question. Why do you think there was no effect of streptomycin? One would think it would be more effective than OTC because it's bactericide. Or would that move the system in the tree? That's a really excellent question from Uta. In the first is the bactericide level. You know, I seems you know, bactericidal and bacteriostatics. Okay, let's tell you the difference. I think is if it's a 95%. If you can kill the bacteria by more than 99.9%, it's called bactericidal. Okay, if you less than 95%, you can kill 95%, but less than 99.9%, it's called bacteriostatics. Okay, they can both kill most bacteria. Okay, I think the reason is I don't know what the reason. I think the let's look at data.
Did you use an injectable formulation, Nian? Or was it just manufactured streptomycin?
The manufactured one. I don't know what the reason. I think one reason potentially is the you see the level of reduction of CLas. Actually, it's not very high. 30 only very short time. Only to 32. The reduction is not as high as than like this OT in terms of CT value tighter, but like 10 time. 10 time. Yeah, I think it's 32. That's how long it can last. Yeah, it still have Liver still there. Still have a pretty high titer. When you look at the Liver in the OTC, seems a little bit better in terms of that in terms of reduce the CLas titer. So we don't know why it doesn't work as well. Yeah.
Any other question or comment?
Doctor, this is Joshua Young, and listening to you talk about the immune response system does remind me a lot of the human health research on gut health and toxicity levels of glyphosate that would block the shikimate pathway from regulating certain minerals needed for disease, the abilities own immune system to function at a high level. And I'm wondering if you had thought about checking different soils for levels of AMPA as a potential toxin that could be in the way of our trees functioning properly again.
That's an excellent comments. Oh, we have not tested that. I think, you know, we are working on a federal grant on the ECDRE. We will probably look different soil type as well. Hopefully, we'll get the funding support on that regard, so we can address your concern. Yeah, I think it's an excellent suggestion.
Another thought is on the Donaldson tree. The big thing, it was better at sequestering calcium, and Dr. Pascoe Avery suggested that if you go back to the early 1900 research and look at the endophytes that were there as parent material, and look at the success we had as citrus growers here, and then look at the trees that we're producing now through tissue culture, and what do those endophytes look like now, and have we lost some of our genetic potential based on just getting away from the true nature of good soil health and good agronomic principles?
That's an excellent question as well. But I think we did some citrus microbiome study, try to manipulate the endophyte, but I think that's why it's not enough. Because what happens is the phloem cell death is really happening systemically throughout the plant. So you might have a little bit, if you manipulate the endophyte, mostly happening is in the root, or you can do a little bit on the leaf. That one is not enough, have enough effect.
Yeah.
So you will economically is making, I did not see any effect.
Thank you. Thank you.
Thank you, Joshua. Here another question from Daniel. How long after OTC injection was the fruit concentration method taken? We taken in like 360 days. So right after the first one. Yeah. So even that's pretty late stage. Really right after the around the harvest, right? So it almost more than 300 days. Okay, other questions? Let me see.
Have you considered SAP analysis as a way to look at nutrition on a little timelier manner and to make better calls in our management system?
Excuse me, Joshua, can you repeat the question again?
Have you considered plant SAP analysis as a tool or a technology to help us manage on a little tighter management system, knowing what the plant needs, maybe even weekly, maybe more foliar sprays of micronutrients, as you suggested? Have you considered SAP analysis as a way to detect those deficiencies a little better than leaf analysis that the system we currently use?
Hey, Joshua, that's an excellent question. What we are doing is we do the phloem SAP analysis every 2 weeks. We want to give you a dynamic data bi-weekly. At the beginning we said monthly, then we change to bi-weekly. I think the dynamic process, you do not need to treat them all the time. As long, I think you several key time points, if we can manage, I think we might be good enough. So we don't have that data yet. Hopefully after 1 year data, we will give you something. What's the best time point we think we can suggest? Yeah.
Thank you.
Thank you. Here's another question from Tall. What was the SWFREC rate in the trial conducted? Thank you, Tom. This is we use the. Okay, let me see. We have all different rate depending on the tree size. Okay, here's the stripe, either this one. Okay, stripe, as 0.5, 1. Okay. Here also here, the trial 2, this little big trees. The biggest, the highest one is 4. I will do small trees. We also do, yeah, 0.25 to 4. Yeah.
That active ingredient or formulation?
This one. This one is the, I think which one? This one we use. I think we use the. This one is the active ingredient. Yeah. Let me get back to you then. I think this one is. Okay, I think that's all the question in the chat box.
Nian, very nice job with the presentation today, and very, I think very useful to everyone.
Thank you, Tom.
Any other question? Thank you all for your participation. Again, if you are interested about CEUs, please send me an email and include your name, email address, and license number. Thank you, Dr. Wang. And thank you all for joining us. And we will see you next month. Thank you, Alison.
You're welcome, Mongi. Thank you, everyone.
Thank you, Mongi. Thank you, Alison. Thank you, everybody.
You're so welcome.
Okay, bye.
We recorded the presentation. We will post the link at the HLB Center website next week. Thank you all.
Thank you.
Bye.
Bye.